Yeah.
So Ruby's based at Imperial. And if you look at the title of this talk, you'll realise this is actually the perfect talk to give at this point in the summer school. We've got multitask, we've got Gaussian processes, we've got Bayesian optimization. So this is going to really hopefully round off everything you've learned. And this work was accepted for publication yesterday?
Yeah,
So
yesterday.
sorry, congrats. Hopefully it's interesting.
Yeah, also you'll notice that the title is actually slightly different to what we're on the schedule. It's a transfer learning schedule and multitask learning here. That's because the sort of framework that we use here can be used for either multitask or transfer learning. So in transfer learning, you tend to have a set of sort of tasks or data sets that you've learned before, and you're trying to use that information to learn a new one. In multitask, you're just learning them all at the same time. We kind of do both of these things, and yeah, the framework can be used for both. So if I say transfer learning or multitask learning, I basically mean the same thing. Yeah, this is the work I did in my PhD. Yeah, recently accepted, which is good. Took a while. Yeah, so first of all, just go over what I'm going to talk about today. I'm going to start off with some motivation, why we did this, the formulation, so turning our problem into maths, then sort of our solution for it, which was this design of experiments workflow. Going to talk about some transfer learning surrogate models, so these are multi-output GPs like you learned about yesterday. Talk about the Bayesian optimization we did, show some experimental results, and then sort of summarize everything. So firstly, yeah, okay, cool. I'll do a picture. Oh, I'm going to get this on there again. Okay, well anyway. So we want to design this diagnostic device. The device itself is quite novel and quite complicated, so I'm going to gloss over it and just tell you the important parts. But we're trying to detect different gene expressions in blood. So when you get infected with a disease, your body responds to the disease, and you'll have different genes which will be up and down regulated compared to a normal person. And we want to sort of detect multiple of these, and each gene, well, it's been proven that for certain diseases, there's certain signatures you get. So we want to detect multiple gene expressions all in one diagnostic, which is going to tell you if you have the disease or not. This diagnostic is PCR based, so the same test that you did for COVID, where you put a swab up your nose. It's kind of based on the same technology as that. I don't know why that figure does that, but in PCR, you are sort of amplifying DNA, and you get a reaction curve that looks something like this. So over time, you're increasing the amount of DNA you have in the mixture. And this is done by repeatedly dividing and then rebuilding your DNA strands. So in our device, we wanted to be able to get an endpoint readout, and we wanted to be able to do this multiplexing of different genes. So we're actually using a type of PCA called competitive PCA. So this is where you have your wild type, or the biomarker from the body that you're trying to detect, and it competes for resources in the reaction with your with a synthetic competitor that we have designed and put in there. By comparing the differences in fluorescence, so each of them has a different fluorescent probe. By comparing the differences in fluorescence at the end of the reaction, we can compare how much of each one we had at the beginning. Now, we want to be able to multiplex this. Multiplexing just means sort of detecting multiple things at the same time. That's because we've got these different genes. So this is for tuberculosis. There's 3 different genes we want to detect. You'll see that one of them is upregulated if you have tuberculosis. So that's what the log odds are, the log odds of having TB. So the first one is upregulated if you have tuberculosis. The second 2 are downregulated, but they're downregulated by slightly different amounts. So I'm not going to go into too much depth about how we do this, but basically by designing our competitors to have different sort of amplification curves, we can combine our outputs in this kind of logistic regression. That's not how to explain to a biologist, but machine learners, it's kind of like you're doing logistic regression, but in your test tube. So this kind of leads us onto our competitor design problem. So for each competitor, we want a specific curve. We don't necessarily, Oh, so it's a synthetic DNA strand that we're putting into the reaction, and it's competing for resources with the biomarker we're trying to. And so that interaction allows us to sort of basically figure out how much we have. So for each of our competitors, kind of going on to the problem we're trying to solve now. So we're trying to design these competitors, these synthetic DNA strands. We want them to have a specific amplification curve. We sort of reduce this curve down to two key parameters. So the rate, which is the rate of the linear part of the curve, we want to adapt that. And then there's also the second kind of nuisance factor that we found when we actually did the experiments, which is that we get linear drift at the end. So ideally, at the end of the reaction, it should just plateau, it should be flat, all the resources are used up, so it shouldn't be able to amplify anymore. In reality, we find that it does, like we get an increase in fluorescence. We don't really know why, but we want to minimize this. So that's sort of our output. So we have two outputs. We want to get as close to the target rate as possible, and we want to minimize the drift. Our input is the strand itself. So when designing the strand, there's the competitor strand, there's these set regions, which are to do with the reaction. So they're set, we can't change them. But then we have this design region in the middle, so we can change what it's made of and how long it is to affect this region. And so this means we turn this sort of very high-dimensional combinatorial problem designing DNA into a really nice, two-dimensional, almost continuous problem of just changing the length of the DNA strand and the GC content. So that's what it's made of. And you can think of GC content kind of as the stiffness of the strand. Yeah.
That might be a really dumb question, but is there a reason you don't just do a keep and
Do we keep or?
so you have like so you're looking for a type of gene expression to have a certain gradient. So I guess you have to have theoretical differences.
What do you mean by gradient?
I like to understand that, for example, the rates.
Yeah.
There. So you must have like theoretical curve comparing to what against that for those measures.
Yeah, I see what you mean. I think we chose to do this because it kind of simplifies it down to just one output parameter, which we're then going to optimize, rather than kind of take the whole curve.
Also, in a QT plot, you can have the Pearson correlation.
We're not too fussed about what the rest of the curve looks like. We only care about the rate. Like, the rate is what matters. And also these are kind of, like, real data is not that ideal. So to get out what this curve is, you have to do some modeling and stuff as well. But that would have been another, like, a potential option. But yeah, so we end up with this kind of semi-continuous space. It's not quite continuous because obviously you can't have, like, I don't know, you can't have 50% GC content of a length of 7, that's not possible. But in those situations where it's not possible to evaluate the point, we just go for like the closest point we can evaluate. And then, okay, yeah, so does anyone have any questions? Yeah.
With that idea, if you mix like two like one like and that work together a little bit, is that you have really never I don't know.
No, that's a good point. So we do mix competitors. Cool. So when we put all the genes and all the competitors in one thing, and this is a whole other presentation. My collaborator has worked on building networks out of these, so you have all these competitors that are all sharing different resources and can build these like quite clever networks out of it. But I don't think that helps with the drift because we don't really know what causes the drift, which is a bit annoying. Yeah.
So you got two objectives, one for the rate and one for the drift, as possible. Do you have like you frame this as like a multi-objective problem? Optimization.
Yeah, sort of multi-objective, more sort of penalised than multi-objective because we put penalty on the drift and we're optimising the rate. But yeah, you have preempted what happens.
And you combine them both into one objective?
Yeah.
Okay.
Yeah. Which actually what my next slide is. So converting this into sort of a mathematical objective, we want to minimize the difference between our rate and our target rate. Each competitor has a different target rate, but that's sort of predefined by sort of some dynamical models. Then we want to sort of minimize this. We want to make sure that the drift is below this threshold. So once it's below a threshold, it's so small it doesn't really matter, but we want it to be below a certain threshold. However, we want to do this for lots of competitors. So for the tuberculosis diagnostic, we have 3. We've also worked on a cancer one, which I think had 7. But there's sort of a whole host of literature of different diseases that all have different numbers of gene expressions. Some go up to like 60. You're probably not going to do 60 in one reaction. But yeah, you can multiplex lots of things. And also we want to be able to take competitors, maybe we've learned competitors for one disease, and we want to be able to think about new diseases using them. So design competitors for new diseases based on the competitors' new diseases. So the way we do this is using design experiments. Design experiments is just essentially using statistics to more intelligently decide which experiments you're going to do. This is our design experiment workflow. So we start off by collecting the data. We use an PCR robot for this. We've got 2 continuous variables, our length and our GC content, and then we've got categorical variables which kind of define the different competitors. That's not too important, but then we use a model to decide what our rate and drift should be. Well, what our rate should be, and we obviously want to minimize our drift, but we decide what our rate should be. We fit a transfer learning model, which I'll go into more depth in a minute, and then we do Bayesian optimization to decide which sort of point to experiment next. So I'm going to talk about the transfer learning models now, our surrogate models for Bayesian optimization. I'm going to introduce 4 models. 3 are kind of benchmark models that we compare against, and 1 is the sort of model that we're proposing to use for this. But before I introduce them, I'm going to justify why we use Gaussian processes, which is maybe preaching to the choir a little bit, but we want to use Gaussian processes because they're good in low data regimes. These experiments are quite time-consuming and quite costly. It takes about a day to run an experiment, but then you have to order new things, so it takes about 2 weeks for the new sequences to arrive. So we want to minimize the number of experiments doing. And they give good uncertainty measures, which obviously is good for data optimization, as Henry told us yesterday. So just some notation, yeah, that's our Gaussian process. For all of this work, we used a squared exponential kernel. We did look at using a squared exponential and linear plus linear kernel, but the linear didn't really do much. It wasn't really necessary with this data. And we use a 0 to mean because we didn't have any reason to use anything else. So the 4 models we sort of consider, going from probably most basic to most complicated. We start with this average Gaussian process, I'll choose in a minute. Then we'll now talk about the multi-output Gaussian process, then the linear model of co-regionalization, and then something called the latent variable multi-output GP. So I think at least the middle 2 were covered yesterday. So the first one, this is the most basic model we could think of, is an average GP. So this is where for each of our competitors, so learning each competitor is a task. So for each of our competitors or each of our tasks, we just fit it all with one GP. We ignore the fact that they come from different tasks and fit it all with one GP. Our reason for doing this is that this is kind of as close to what an experimentalist might do in their head as we could get with the Gaussian process. So if you're an experimentalist and you're trying to design a new competitor and you're trying to find a more optimal region, you're probably just going to experiment in regions that have been optimal in the past for past competitors. So that's why we use this model as a benchmark. And this can also be thought of as total transfer. Like you're transferring all the information from all of the competitors to all of the others without sort of leaving out irrelevant information. The next one we'll talk about,
could you elaborate a bit on why there are different competitors that can take different tasks?
So they're all like, so the different competitors will have different, they will behave differently. So for different lengths and GP content, they will have different rates. But we believe there will be some similarity. So like some regions of the space we know will be higher, some will be lower. So that's why we want to use transfer learning. Does that answer the question? Yeah, because if they were going to be completely different, then transfer learning wouldn't help. If they were exactly the same, then you would just use like one UP. So the next UP, the SIDRs, the multi-output GP. This is essentially where you have this sort of block diagonal covariance matrix, and you essentially this is assuming that all of the output functions have the same kernel and the same kernel hyperparameters, but no information is transferred about function values. There's no correlation between your outputs. We chose this as like fairly close to just not doing any transfer learning at all. So this is, there is the shared hyperparameter thing, so you could argue that there's some information transferred, but in general, like this is no transfer learning. The next one is the linear model co-regionalization, which again I think was covered yesterday. I'm not sure if it was called this yesterday, but essentially this assumes that all your output functions are a linear combination of a small number of latent functions. So say we have P output functions, then our P output function is a linear weight of some latent functions. Normally you have less latent functions than you have output functions. And then you also allow it to have some independence by allowing it to have its own little latent function to deal with that. And you end up with this kind of covariance matrix where you do have correlations between outputs. I think this was covered yesterday. And we're using a special case of this called the intrinsic model co-regionalization, which is basically you assume that all your latent functions have the same kernel. So you could have different kernels for your different latent functions, but it'd be the same one. And if you're interested in it, Horatio has a really good sort of review paper about this, which I would highly recommend if anyone wants to learn more about multi-output GPs. Any questions so far? They're sort of our three sort of baseline comparison ones. Okay, so the downside of the linear model co-regionalization sort of has two downsides. The first thing is it assumes that the sort of similarity between your functions is linear, which might not be the case. We might want to be able to model more complex similarities. And the second thing is that your weight matrix, your W, is deterministic. So that can often lead to overfitting, particularly in low data regimes. And because we're using experimental design, we don't have that much data. So our sort of proposal for the way to go around this is to use something called the latent variable multi-output GP, which is on this paper here. And this works by you assume that your output is a function of both your observed input, so your length and your GC content, but then also a latent variable. And each output function gets its own latent variable. I think the best way to show this is actually look to the image. So this is sort of what you end up with. So each output function has its own latent variable, which you learn on a latent space. And then you also learn what the function is as well. And the nice thing about this is that you can do automatic transfer learning. So the model should learn, by placing the different functions at different points in the latent space, it will sort of learn how similar functions are. So two functions that it thinks are really similar will put next to each other, and two functions that it thinks are really different will put really far apart. And then based on the distance you have the sort of correlation, so it can decide which functions to learn from and which functions to exclude. Yeah?
So is there some kind of norm? So
the latent space is a GPSL. So yeah, it's just got a kernel. So it's got the kernel on the latent space. So it also learns the length scale and latent space as well.
Is it always 2D then?
No, you can use any dimension, and because it's Bayesian, you can it can automatically determine what the dimensions of the latent space should be. So really, what you should do is have quite a high dimensional latent space, and then sort of it will prune it itself. I've used 2D here because you can plot it nicely.
How do you train the dimensional latent space?
How do you train it?
Like how do you learn the best latent space?
So because it's a GP and it has the length scales, what you can do is you like the hyperparameters you put in something that is much higher than the dimensions you think it is. So I think in the first version of Bayesian we just did 2 dimensions, and then we increased it to the second version. So we did I think like 10 dimensions and let it prune itself. And because it's a GP, it can just learn like a long length scale on certain dimensions if you use the right kernel. So if you use a separable kernel, so a kernel where you assume all the input dimensions are independent, then it will just learn a really long length scale for the ones that are irrelevant, and then a shorter length scale for the ones that are relevant, which is quite nice. Yeah.
Is it easy to do? Like really, yeah, I can look at a recorded like it looks to do one of the places you want, like before.
Yeah, so
That, or have
I don't know how much difference it makes going from like 2 to 10. It's really difficult, like a one-dimensional latent space. It's hard because points can't move around each other, so you can often end up with sort of issues with that. So if you have a one-dimensional latent space, you have to make sure that they're properly ordered first. But one of the downsides of this model is the initialization and the optimization. So we initialize this it's a bit complicated, but you initialize it by sort of fitting a functional PCA, and then you fit a Bayesian GPRDM on the output of that, and then that's your initialization for the latent space. Yeah.
So basically what you do here is you have an input, and then you transfer the input into latent space using Gaussian process, and then from latent space you
So the latent space is like, you can think of it as adding extra input dimensions. So you don't take your input and put it in your latent space. It's more like we had a 2-dimensional input, we now concatenate 2 extra dimensions, but those dimensions are unobserved. They're our latent dimensions.
how so can we just use the PCA again and concatenate our data?
For initialization, yes, basically. I mean, it's a bit complicated because each function has its own latent, so you have to make sure that all data points in the same function all have the same latent point. Does that make sense? I don't, yeah. Yeah.
Maybe that's a good question. So is there any close solution to this multi-output?
No, you have to use variation reference to train it.
So it's the non-Gaussian.
Yeah. I think someone mentioned variation reference earlier in the week. Yeah, the same thing. Also the same kind of techniques that you use for sparse GPUs. Yeah, which again makes training a bit harder.
What about the likelihood? So we use the algorithms of the output, the likelihood of the output, the likelihood function.
What do you mean by the aggregate?
I mean, so suppose we have like 4 outputs. So for each output, it has like it's sort of like, I mean, if we use Gaussian process on one output, it has like one likelihood function. So we will have like 4 likelihood. We just aggregate it.
So we actually just use this as a single output thing. We just fit 2 for our 2 different outputs. Our output being our rate and our drift. We could have built more into one model, but we thought that was too complicated, so we just keep them separate. So this is going from from 2 observed inputs plus however many latent inputs you want to 1 output.
Basically, there will be like, if it's 4, there will be 4 Gaussian process.
Oh yeah, yeah, okay, I see what you mean, yeah, yeah. Yeah. I don't know what you mean by the likelihood thing yet, but...
Yeah, I mean like to calculate it. I mean the point of the question is, will you at the end, will you have like 4 independent Gaussian process, 4 separate models, or one model that can directly predict the 4 outputs?
So we have one model, and then say I want to predict on function 4. What I do is I go to that point in the latent space, so I fit my latent space variable, my latent variable to be whatever that is, and then I predict on function 4. Yeah, which also is one of the benefits of this model. It means you can sort of imagine what unseen functions would look like, what unseen surfaces would look like. So this is something we actually do. So we have our competitors that we've designed, and we want to design a new competitor. We have no data on that. We look at competitors we've designed before that for some reason are similar, and then we take a weighted mean in the latent space, and we use that to predict our new unseen surface. We didn't do like a huge amount of investigation into which similarities matter and which similarities don't, but we found it did improve the prediction on unseen surfaces, which is good. Yeah? You just go to the latent. So if you want to predict on it, you've already learned what your latent variable is, so you just go to that latent point, that point on latent space and predict from that.
Okay, but when you train that's your output.
Yeah, so this is actually, yeah, it's actually like it's more like, so we have, so say this one here, we have 1 input dimension and we have 2 latent dimensions. So we actually have a 3-dimensional GP. It's just that 2 of the dimensions are latent. So then so you just train it as if it's a 3-dimensional GP. But then when you want to make a prediction, you sort of condition on your latents and then predict from that. Yeah, that's maybe a better way of saying it. Yeah? So the way you get them. Yeah, it is quite it's very close to the initialization. I don't know if anyone's familiar with the Gaussian process latent variable model, but it's very connected to that, and all of these sort of latent variable models are quite sensitive to initialization. Yeah?
How do you measure the proximity between latent variables and especially average general based on the variance what measure do you use to measure the proximity between
we don't actually need to measure the distance because it's a GPU. We just let the GPU do the work to figure out what the variance is between them. Yeah. But if you needed to,
yeah, because this is illustrate that functions with similar functions are more likely to aggregate all the data. I'm just curious if your main is level aggregating this which
I guess you could use anything. It kind of depends. So like you could just use any I have seen people do these kind of things and then do some kind of clustering on the latent space or things like that, so you could do that. Yeah, any other questions about this model? So this is just a summary of our 4 models that we're looking at, going from sort of simplest to most complicated. So we start with, the average GPU, the MOGP, the LMC, and the linear, the LVMOGP, going on after 4. And you can see how how the predictions differ. So like the average GP is just predicting the same thing for every function regardless of the differences. The MOGP shares the kernel hyperparameters, but sort of on the bottom one where it hasn't seen much on the right hand side, it's just gone through prior. And then the LMC and the latent variable output GP is able to predict a bit more. Okay, now moving on to the sort of Bayesian optimization part. So we have our surrogate model for our different competitors, or DNA strands. We want to optimize them now. And we sort of are doing a sequential optimization where we're optimizing, we're doing a sort of sequential optimization. So we're going to use Bayesian optimization. This is my slide on what Bayesian optimization is, but Henry did a good introduction yesterday. Just a little reminder, we have our surrogate function, and then we have our acquisition function, and we just maximize our acquisition function to choose which point to do next. So looking just at the rate optimization for now, we've joined the drift part, we're just trying to optimize the rate. We want to minimize, so normally in Bayesian optimization, you're either maximizing or minimizing the function. However, we want to minimize the difference between the rate and the target rate, which is a bit harder. But luckily, someone had already done this, so I didn't have to figure out how to do this. And essentially what this paper is doing is they define and use a stochastic variable, which is just the difference between your bars, our rate, and our target rate, the squared difference. And then they redefine the expected improvement basically in terms of like chi-squared distributions rather than normal distributions. So you can see that it's like quite similar to expected improvement. We still have our, we're still looking at our minimum value, and then we use an approximation to chi-square distribution. It's a chi-square distribution because that's the square of the normal distribution, which you have to do when you take do the difference. And so yeah, this is how, yeah.
So you're still modelling like F, you're not modelling F squared or anything?
Yeah, so it's still modelling F. The problem with modelling F squared, which you could do, is that that's quite hard to do with GP, because then you can't go negative. So maybe if you didn't expect it to, if maybe you didn't expect your GP to copy access very much, you could do that, but it wouldn't work for us. Yeah, but this is like a nice workaround for that. But then, remember, we've got this drift term that we want to penalise, and we want it to be below a certain threshold. To do this, we use probability feasibility. This is actually generally used in the constrained optimization literature. But basically, this is just the probability that our drift is below a threshold. And to create our final acquisition function, we just multiply this by our expected improvement. To be honest, there's definitely better acquisition functions we could have used. And if I had another year or two on my PhD, I probably would have explored them. But we just needed something that would work. And we looked at various different things. I spent some time looking at constrained optimization. The problem with constrained optimization is we do want to allow it to be above the threshold sometimes. So for some competitors, the drift is just always high. It's never below the threshold. So we would never get a feasible point. And for other competitors, we sometimes get, sometimes choosing a point that, just got a little bit too much drift gets a much better rate. This is a scenario when a kind of multi-objective thing would come in handy, like the Pareto front stuff that Henry was talking about yesterday. And then we'd also have to go back to our experimenters and figure out, what is an okay amount of drift and what's too much, what's the trade-off between your rate and your drift. So if we were going to do this really rigorously, that's what we would have done. But we found this work well enough for our scenario. Anyone have any questions about the Bayesian optimization part? Yeah.
So just check that did you play around with weighting those two modified aspects of the vehicle?
No, we didn't, and again, that's something that I would have done with more time. The thresholds were set by our experimenters with designing the system, so I was fairly happy that the thresholds were right. But that was partly our problem because you normalise it, the drift and the rate have very different magnitudes, and then you normalise them both, and then actually what is the correct, what is the combined one, really. But yeah, we settled on this one. So yeah, going back to just a reminder of what our workflow is. So we did try and do this in sequence. So we were doing the experiment and doing the surrogate model, doing the Bayes-Opt, and then selecting the next point. However, this didn't really work to plan for various reasons. Some issues with experimentation, some issues with modelling, and then my experimental collaborator left. So we unfortunately weren't able to do this all in sequence. But just to sort of prove whether our method worked, we did a kind of retrospective thing. So we did end up getting quite a lot of data in the end, but we couldn't really say that we'd done data optimization to acquire it. But we are sort of using the data set that we had from that, and both just the experiments that my experimenter was doing to do his own work. We did end up with quite a reasonable big data set. I think it was about 500 data points on 32 different competitors. So we sort of retrospectively analysed our method based on this. So the first thing we did was cross-validation. I have to look at my notes to see exactly how we did this cross-validation. So what we did here is for each cross-validation run, the training set consisted of 2 fully observed competitors, so just all the data from the 2 which had the most data on, and then a random subset of the remaining data. And this was repeated for different percentages sort of the data being in the training set, and for each percentage it's repeated 200 times, so 200 different splits of the data. What we see from this, and the metrics, what this plot is actually showing. The top one is the root mean squared error, so between the mean and the true value of the data point. The bottom one is the negative log predictive density. So this is a good measure that tells you not only how good your prediction is, but also how good your like uncertainty estimate is. And what we found from this is that the latent variable multi-output GP fits the data better, which is good. That kind of confirms our hypothesis that we needed something a bit more complicated than the LMC. You'll also see that on the bottom one, the LMC, so the blue sort of dot dash line, has really bad NLPD at low data regimes. This is something that we found both with this data set and with our synthetic data set, was that it really overfits at low data regimes. And this is because that W matrix, that similarity matrix, is deterministic. So it has no way of expressing uncertainty in what the weightings of these latent functions are. Yep.
Sorry,
Linear model of co-regionalization. So it's the one where you have this like linear combination of some latent functions. And then sort of the fact that the latent variable multi-output GP outperforms the other methods shows that some transfer learning is useful. So that's also good. Our competitors aren't all just completely different. Another nice property, which I kind of touched on earlier with some of the questions, is in the latent variable multi-output GP, it learns the latent space. So this is what the latent spaces it learn. So it sort of scattered them all, across the latent space. The one that's like the most interesting here is this drift one. So here it has done the thing where it determines what the dimensionality should be. So it switched off the second length scale and just only used the first one to explain the data. So that's really nice. The second interesting thing here is that the X's and the dots are competitors with different fluorescent probes. So we didn't really know this sort of before doing the experiments, but actually the fluorescent probe makes a big difference to what the drift is by like an order of magnitude. And what it's done here is it's separated out apart from this rogue like orange X on the edge, it has it's separated them out by what their fluorescent probe is. So that also shows that the latent space is learning something. So that was a nice result to see. Then moving on to our Bayesian optimization experiments. So here we did a sort of retrospective Bayesian optimization. So we separate out the data into a sort of training set, and then each model is only allowed to choose points from a pre-existing data set as the next point. We investigate two different scenarios of choosing your first point. The first one was all the models just see the most central point to begin with. The reason for this is just the closest thing that an experimenter would do. It also reduces the variance in your space the most. The second one was that we let the model choose the first point. So for the average GP and the latent variable multi-output GP, this is doable because they can both make predictions on new surfaces. The linear model co-regionalization and multi-output GP can't make predictions on new surfaces. So for those ones, they just choose a random point. And then we explore two different learning problems, which roughly correlate to transfer learning and multi-task learning. So the first one is where so for both of these problems, we have two fully observed surfaces already. And then for the first one, we learn all the other surfaces at the same time, so adding 1 point to each surface each time. And then for the second one, we learn one surface at a time with all the other surfaces being in the training set. So this kind of relates to this, the first scenario is where we're learning lots of competitors at the same time. Second one is maybe we've learned lots of competitors for different diagnostics, and we want to design a new diagnostic, so we're just designing one or two new competitors in one go. So here, I've plotted the cumulative regret as we go against a number of iterations. Our first column is single objective, so that's only optimizing the rate. And then the second column is with the drift penalty. And this is the scenario where we're learning lots of surfaces at the same time. And the shaded region indicates the upper and lower quartiles for 24 different seeds. So the sources of variation here. In the bottom one, you see these sort of big bars for the MOGP and the LMC. That variation has happened because of the difference in starting point, because it's just randomly chosen. And then with the other models, you get variation just due to the optimization. So because particularly the latent variable multi-output GP is quite sensitive to initialization and the optimization procedure, you get some variation in the fit based on that with different random seeds, which is annoying, but that's what happens.
So, can I ask a question on that? So it looks like, the last model, the one NEMOTP.
Yeah, I didn't name it.
It looks like it's very, very small variance on over the runs. But then you said it had a lot of variance, or did I misunderstand that? Because it looks fantastic, right?
Yeah, so it's not too bad here. And also, it doesn't have a lot of variance here, but it is annoying that it is dependent on the initialization.
Yeah, but that's
but in theory,
people, right?
yeah,
it's
in
like
theory it should always be the same.
fantastic,
Yeah.
right?
But we would expect it to, because this is with, yeah, actually no, don't worry, ignore what I was about to say, yeah.
But then you get a little bit.
Yeah, you get a little bit there, yeah.
Yeah, exactly. And it's kind of interesting because the others don't, right?
Which is maybe like, well, actually, the multi-output GP does, the LMC does a bit in the drift one as well.
Yeah.
It's maybe not so clear up there.
And it was interesting because that's what has such a bad fix, right? Which you would then think, if I give it random initialization, you would just follow up because it continuously overfitting, right?
Yeah, but the overfitting is often like the same. It is getting into the same local optima. So I think it's partly because those models, like the average GP, the MOGP, and LMC, like the optimization of it, they're all just using the marginal likelihood and optimizing it. They don't need variational inference. And it's just an easier optimization problem.
Yeah. So it's consistently wrong, right?
Yeah.
In some sense.
And with all the models, so because we needed to do random restarts for the latent variable multi-output GP, so that's just where we initialize it with different things, run it, and then we pick the one. Luckily, you can use the elbow to pick the one. You just pick the one with the highest elbow or the highest marginal likelihood. So that's good. But because we were doing random restarts for that, we felt we should do random restarts for all of them, to be fair. But actually, it didn't make much difference for the others. So all of these have random restarts.
How big is your random design? Like your starting of it, like how many points do you pick in the initial?
So we did, I think we do like 9 random restarts for each one.
Then how was how big was the set that you started with for one random?
Big, because
Okay.
so the latent variables we initialize in different ways. So sort of 3 different ways we initialize them. But then it's the inducing points that I actually think make a big difference. And that's just, I think we just did a random subset of the data. So it's sort of how much, whatever permutation of the data. And then we also did random length scales and variances as well. So it, even 9 is not covering the space at all, really.
No, but it's cool that I think it's very cool that it's very stable.
Yeah.
On this experiment.
Yeah, on this experiment, it did quite well. So what they've shown in general is the latent variable multi-output GP had less, regret overall. The models that can choose their first point tend to do better. So that's actually like a really good bonus of this model, is that you can choose the first point well, which, everyone knows, well, not everyone knows, but like it's general knowledge that like in Bayes-Opt that makes a huge difference if you start in a good area, a good region. Is there anything else I want to say about this? Yeah. Then moving on to the sort of one surface at a time things. This is where all of the surfaces are in your training set except one, and then we're learning that one. And this is just sort of averaged over all of them. We can see again the latent variable multi-output GP does better. You can also see the LMC does a lot better. Oh yeah. Choose it. Sorry, say that again. So with Henry's probably better better to answer that question. So here we just did it based on getting the model to choose where to start by predicting on an unseen surface. But there's lots of different things people do in general. They might look at previous experimental things and start in the regions that were optimal before. There's all sorts of things. There's like a whole host of literature about how to do that for Bayes-Opt. And here we see that the linear model of co-regionalization does better. That's because it's got more data, we're not getting this overfitting issue so much. So it can do better here. But in general, we still see that the transfer learning is helping. Any questions about the results before I just go on to a summary? Yep.
Do you have an intuition as to why the graph on the right tends to have a higher
Because the drift was, because it's kind of a harder problem, and actually something that we found in general is the models for the drift just weren't as good as the models for the rate. So this kind of, these kind of multi-output models actually modeled the rate pretty well, but the drift was difficult because you had this sort of order of magnitude difference in it. So I think sometimes the drift penalty was actually harming the optimization because it would think that a certain region was bad when actually it was fine. So I think that's why it's worse. So in summary, we sort of converted this DNA design problem into a continuous optimization problem and a transfer learning problem. We compared a number of multitask GP learning surrogates and found that the latent variable multi-output GP had the best predictive accuracy and did best in Bayes-Opt. And these results show that this method can be used for generating these new competitors. So I'm actually not sure how much he uses this, but my experimental collaborator who I worked on this with has now gone off and started a startup that is doing exactly this, building these diagnostics. And so I think he is using this sort of Bayes-Opt approach to design new competitors. Possible extensions or things that I wish I could have done if I had more time. are sort of better exploration of the acquisition function. Something that I think could be quite interesting is a sort of transfer learning acquisition function where you take into account the fact that you're learning multiple surfaces at the same time. So maybe if you think two surfaces are really similar to each other, there's no point in picking the same point on both those surfaces. You could sort of learn more information by picking different points. Again, it kind of may be a bit connected to the batch optimization that Henry was talking about yesterday. We can improve the models for drift. This might even just be sort of separating out depending on what fluorescent probe you're using. Something that I really liked was the fact that in the latent space you get this separation and it can choose the dimensionality. And I'd really like to investigate whether it over prunes or under prunes the latent space. Like, does this latent space actually have meaning, and is it choosing the right dimensions based on the marginal likelihood or the elbow? And then the main thing is the variational inference of this latent variable multi-output GP, which is true of all latent variable GPs. They're all trained using variational inference. I think they're really like cool, powerful models that can learn a lot, but also the training and the optimization of them is a bit difficult. And so I think, yeah, some work on that and how to do that is needed to make these like really applicable, particularly to people like in industry. Yeah, thanks for listening. Here's a link to the paper and the website.